Hello everyone, this is your educator Ria from Biotecnika. So today we are going to learn about auxin phytohormone. So this auxin is derived from the term meaning to grow. So this is the first growth promoting substance that were observed in the plants that modulate the plant growth by promoting the phenomena of cell elongation. So under this topic, you should be very clear about the biosynthetic mechanisms, the signaling pathway of this phytohormone, what is the functional profile, discovery, as well as the transport mechanisms and the related inhibitors to it. So from this topic, you can expect questions in every competitive exams like CSIR NET, DBT, GATE, ICMR and others. So let's dive into the session and understand the concept of auxin phytohormone and all the important points related to it. So firstly, we'll understand the introduction of auxin phytohormone. So this auxin phytohormone, auxin meaning to increase. As I've already mentioned, it is derived from the hormone and it was discovered in the tip of the plant body. So they are the derivatives of indole compounds. So shall we say this indole compound are the precursor of this auxin phytohormone? Yes. So they are the precursors of auxin phytohormone. Now let's see the types of auxin. There are two types of auxin. The first being the natural auxin and the other is the artificial auxin. Natural auxin is the one that is synthesized in the plant itself, whereas artificial auxin that is not synthesized in the plant body, means it is synthesized or generated outside in some kind of laboratory or industry. Now, it's very important to understand and learn the example of natural auxin as well as artificial auxin. So now let us see the examples of natural and artificial auxin. So if you see under natural auxin, you have to learn about three major examples. The first being indole 3 acetic acid, the second is indole 3-butyric acid and the third is phenyl acetic acid. Whereas under artificial auxin, you have to learn 5 major examples. The first is 2,4-D, which also acts as an active herbicide. Herbicide means it is responsible for the killing of small herbs. So what is the extension of this 2,4-D? It is 2,4-dichlorophenoxyacetic acid. Sometimes in ICMR exam, they focus on the extension of this 2,4-D. So it is 2,4-dichlorophenoxyacetic acid. The second being naphthalene acetic acid or NAA, which is used as a rooting powder. Now suppose if you are doing any kind of plant tissue culture and in an experiment if you are adding this NAA, so it will induce the formation of roots in the plant body. So this naphthalene acetic acid is an example of artificial auxin. The third being dicamba, which is again the active herbicide. Now what is the extension of this dicamba? It stands for 2-methoxy-3,6-dichlorobenzoic acid. It is 2-methoxy-3,6-dichlorobenzoic acid. So again you can see it is an active herbicide that is used for the killing of herbs. The fourth being picloram or tordon. So this picloram is also known as 4-amino-3,5,6-trichloropicolinic acid or tordon, or you can say picloram. So again this is an active herbicide and the last is 2,4,5-T, that is 2,4,5-trichlorophenoxyacetic acid. Now here you have to learn about the active herbicide example. So they are 2,4-D, picloram as well as dicamba. So this is all about the types of auxin. Now let's see the discovery behind this auxin phytohormone. So the major candidate for the discovery of this auxin goes to Charles Darwin, who discovered the phenomena of phototropism on a 4-day-old seedling in 1880, where he explained that Auxin phytohormone or some kind of growth stimulus, it is produced in the tip of the plant that is transmitted to the growth zone and that causes the cell elongation. Now let us understand this with the help of diagram. So suppose this is a coleoptile tip. So when this plant is exposed to a continuous sunlight, so what it will do? It will produce some kind of chemical in the tip of the plant. Now this chemical is transmitted to the growth zone. Suppose some kind of chemical is produced which is transmitted to the growth zone. Now once this is transmitted, it will cause the elongation of the cells. So what will happen? Suppose the growth substance is produced here. It will be transmitted to the growth zone which will cause the elongation of the cell and in turn results into the bending of the plant towards the light. So this is what called as phototropism. Tropism means movement. Photo means photon in the presence of light. So he proposed that some kind of growth stimulus is produced in the tip of the oat seedling that is transferred to the growth zone and causes the cell elongation. This is only causing the bending of the plant towards the light and this is what called as phototropism. So this work was further extended by another group of scientists, that is P. Auxin and Jensen in 1913, where they explained about the stimulus of auxin in plants. So they informed that if some kind of growth stimulus is produced in the tip of the plant, that will be transmitted only if there is a water permeable barrier in between. And if there is a water impermeable barrier, there will be no transmission of this growth stimulus and no phototropism will be seen. So what they did, they took gelatin as well as mica as a experimental material, where this gelatin is a water permeable block. That is a water permeable compound, where this is water impermeable. That is impermeable to water. So what they did, they removed the tip of the plant. Suppose this is a coleoptile tip. So they just remove the tip of the plant. Now once this is separated, this is a coleoptile stump and this is a tip. So they just place the gelatin block in between. So gelatin, it is a water permeable that will allow substances to pass through it. So suppose if the plant is again exposed to sunlight, again growth stimulus will be produced in the tip of the plant and this growth stimulus will again be transmitted to the growth zone. And again that cell elongation will happen, ultimately bending of plant towards the light. This is what called as phototropism. So they just propose that if water impermeable barrier is there, there will be no transmission of growth stimulus. Where if we place any kind of gelatin or agar block, so that transmission will happen and there will be a phenomena of phototropism or the bending of plant towards the light. So here bending was seen towards the light source, whereas in case of mica, no bending was seen. So this was all about the discovery of auxin phytohormone, where Charles Darwin proposed the phenomena of phototropism, which is very important. This was further explained by P. Auxin and Jensen in 1930. So now let's come to the biosynthetic mechanism of this auxin phytohormone. So I've already mentioned that they are the derivatives of indole compound. So here tryptophan amino acid, it is acting as a precursor for the synthesis of auxin phytohormone. Now what are the site for the synthesis? It is shoot apical meristome, young leaves and young fruits. They are the primary site for the synthesis of auxin. You have to remember about SAM, young leaves as well as young fruits. Now there are two pathways for the synthesis of auxin. The first is tryptophan dependent pathway, as a pathway that requires tryptophan amino acid for the synthesis of auxin phytohormone, and the other pathway is tryptophan independent that doesn't require any tryptophan precursor for the synthesis of auxin. So this pathway, that is tryptophan independent pathway, it is seen in the mutant form of maize as well as Arabidopsis. Now coming to the regulation of this auxin phytohormone. So what happens? This auxin is required for the growth and survival of the plant. So once this auxin is synthesized at higher concentration, it is toxic for the plant cells. So it has to be maintained at the optimal concentration. And if the homeostatic control is not there in the plant body, it will build up to toxic levels. So what are the different mechanisms how auxin is regulated inside the plant cell? So there are three main mechanisms for its regulation. The first is the storage mechanism, where the auxin is conjugated to some type of other molecules and it is stored inside the plant cells. And now it is in its inactive state. So it will not be toxic for the plant cell. So first is the storage mechanism. The second being the degradation pathway, where the auxin that is synthesized in a larger amount is degraded into some other constituent. Like firstly, it will be conjugated to the hexose sugar and further catabolism of auxin hormone will take place. And the second is the oxidative degradation of this auxin phytohormone, where it is degraded and the permanent removal of auxin will be taken place in the plant cells. So under degradation, that is catabolism, we have two major pathway, that is conjugation as well as oxidative degradation. Conjugation with hexose sugar causes the auxin to be degraded in the plant cell and make it inactive, whereas oxidative degradation causes the permanent removal of auxin from the plant cell. And the third is the inactivation pathway, where this auxin is conjugated to some other type of amino acid. And now what happens? This auxin is inactivated. If this is inactivated, it will stop all the signaling related to the auxin phytohormone. Now this is inactivated. So there are three basic mechanisms from which we can just stop the synthesis of auxin phytohormone, or we can just make it inactive. Now coming to the signaling pathway. So this auxin that is synthesized in the plant itself is acting as a ligand for this signaling mechanism. So what is the ligand for this pathway? Auxin itself. What is the receptor? That is transport inhibitor response 1, or you can say it in a short form, that is TIR1 or AFP125. So these are the receptor for the signal that is auxin. So this TIR1 is a F-box protein. This is a F-box protein. What is the function of F-box protein? It will cause the ubiquitinylation of the specific target and mark that protein for 26S proteasomal degradation. So what is the inhibitor involved? It is AuxIAA. Transcription factor is auxin response factor, that is ARF. What is the signaling pathway? That is ubiquitin mediated 26S proteasomal degradation pathway. So what happens when no auxin is present? That is one of the example of auxin is indole 3 acetic acid. So you can see here no auxin is present, that is minus IAA. If no auxin is present, it will not bind to its receptor, that is TIR1, and this receptor binding sites are free. So what will happen? This inhibitor will bind to the transcription factor. So you can see the inhibitor AUX/IA, it is binding to the auxin response factor and is stopping the transcription of the genes. So there will be no auxin inducible genes expression. But if the auxin is synthesized in the plant cell, so you can see auxin is present in more amount. So what will happen? It will occupy the sites for the receptor, that is TIR1 as well as AFB125. So here in the diagram, it is mentioned about TIR1. You can see here two auxin molecules are bind to it. So what will happen? This TIR1 is a F-box protein that will add the ubiquitin tag to the target. Now what is the target for this receptor TIR1, that is a F-box protein? It is AUX/IAA, which is an inhibitor for the signaling pathway. So you can see here, this TIR1, it will send or add the ubiquitin tags to this target, that is inhibitor, that will ultimately mark the protein for the 26S proteasomal degradation pathway. And here you can see the protein, that is inhibitor, is degraded. Now the transcription factor is free, because you have seen this inhibitor is binding to the transcription factor. So as to free this transcription factor, it is very necessary or important to cleave this inhibitor. Now the inhibitor is destructed or it's cleaved. So ARF is free, which is a transcription factor. It will bind to the DNA, that is the auxin response element, and will cause the transcription of genes, and hence the auxin inducible genes will be formed inside the plant body, and this will only show the auxin induced gene responses or functional profile of auxin in the plant cell. Now let us see what are the functional profile of auxin phytohormone. Now this auxin serves many different functions in the plant body. The first being cell elongation and expansion. As we have already seen during the discovery that some growth stimulus was produced in the tip of the plant that was transmitted to the growth zone and causing the cell elongation. So the first function is cell elongation as well as expansion in the plants. The second is the vascular differentiation. That means the formation of vascular components like xylem and phloem. So if there is a high concentration of auxin in the plants, so it will induces the differentiation of xylem as well as phloem. That means both vascular bundles will be formed. But if there is a low auxin in the plant body, it will only induce the differentiation of phloem, whereas no xylem will be formed in the plant. It also acts as a herbicide, as we have seen in case of artificial auxin like 2,4-D, dicamba and picloram. They were acting as an active herbicide so as to kill the herbs. It also helps in the parthenocopy process. Now what is this parthenocopy? It is a process which induces the development of flowers and fruits without fertilization. Now if you want to induce the promotion of flowers and fruits, that too without fertilization, you have to provide IA, IBA as well as NAA from outside. So both these IAA and IBA, they are the examples of natural auxin, as well as NAA, that is naphthalene acetic acid. It is an example of artificial auxin. Now in which plants you can promote this parthenocopy? It is pineapple, banana, cucumber, grape, orange as well as tomato. Now from here you can expect an experimental based question where they will design an experimental question in part C and they will mention one plant out of all the examples like pineapple, banana, cucumber. They can take any one example and they will design an experiment where they will mention 4 phytohormones and you have to select one phytohormone out of all. So if there is a name of pineapple, banana, cucumber, grape, orange or tomato in the question, then there you have to select the auxin phytohormone, because in all those plants parthenocopy is induced by this auxin phytohormone. Examples IAA, IBA as well as NAA. It also helps in the process of gravitropism. Now what is this gravitropism? It is a growth of the plant in response to gravity, which enables the root of the plant to grow downward into the soil and the shoot of the plant to grow away from the soil. So in this way, gravitropism is seen in the plant with the help of this auxin. Now this gravitropism is one of the critical steps during embryogenesis, which is taken care by auxin phytohormone. It is also helping as a mobile chemical signal during the embryonic development of the plant, and thus it is also helping in embryogenesis process. So we have seen gravitropism as well as embryogenesis. And the last being apical dominance. What is this apical dominance? It is a process in which the apical bud of the plant is dominated over lateral bud. So means this central stem of the plant will grow, which will inhibit the sideways growth of the plant. And that is why it is called as apical dominance, where the apical bud is dominated and inhibiting the growth of other lateral buds. So if there is a high production of auxin in the plant, only the central stem will grow, because here you can see the phenomena of apical dominance. So whenever the gardener they does any kind of pruning or they remove this tip, then only you can see the sideways growth or the hedgehog growth of the plant. This is because of the apical dominance phenomena. So this is all about the functions of auxin phytohormone, where we have seen the cell elongation and expansion, vascular differentiation, how it is acting as a herbicide, parthenocopy, gravitropism, embryogenesis as well as apical dominance. So the last part is the transport of this auxin phytohormone. How auxin is transported in the plant cell. So once the auxin is synthesized, it is transported in the plant cell with the help of the polar transport. Now there happens a polar transport of auxin which is responsible for the transportation of auxin to other parts of the plant. So we have seen the site of synthesis, which is the young leaves as well as the shoot apical meristem. So suppose if any auxin is produced in the SAM area, that is shoot apical meristem. So with the help of polar transport, it is transported to other body parts of the plants. So what are the protein involved in this transport? So there are two types of protein that are involved in the polar transport of auxin. So the first being ATP protein, which is the integral protein. It stands for ATP binding cassette protein, and the other is the PIN protein. So from here you can expect question. PIN protein is very important, which is responsible for the efflux of auxin. So it helps in the polar transport of auxin. This polar transport of auxin was for the first time studied in maize. Now what are the inhibitors of this transport? It is NPA, it is PBA as well as TBA. So you can get the question from the inhibitors related to the transport mechanism. So the first inhibitor is PBA, which is pyrenoyl benzoic acid. You have to remember the names of these inhibitor. The second being TBA and the third is NPA, that is naphthyl thalamic acid. So this is all about the auxin phytohormone, where we have seen the introduction, biosynthetic mechanism, types of the auxin phytohormone, what are the discovery behind this, what is the regulation of this pathway, signaling mechanism, what is the functions as well as the transport. So I hope this topic was clear to all of you. Thank you for watching this session. Meet you in the next video. Till then, take care. Bye bye. Have a nice day. Happy learning.